Brother, your balance is done. Love and light. Love and light, everybody. Mr. Jimmy Schumacher. This make tour. Be helping sister and her son. She collects cats and stuff for the city, for the business, non-profit. And she helped a family that those people just came into contact with people from off the cruise ship. And to help our friend, help our family and community, and show and prove what we know and what we can do for people. Get this across the world. Fuck the corona babies and shit. Living with this shit forever like the little fucking whack job ass quiche states. Going for the throat and killing this thing. Love and light.
You're just gonna make sure that the bottles are really dry. So if we do it with the funnel, it would be better to put the labels on.
Brother Jeremy. Almost updating everything. I wasn't able to do that.
His wife's picture.
She's gonna be putting your picture in. I sent it just to your name. So let me know it helped you what I sent you. Love you, brother. Got this going. Now we got this thing. Get these guys filled up. Send you in sister's piece. And start infusing actual 1 cups. But it's not 1 cup. It's a cup that makes all kind of shit. And it's actually a cup of life, right? It's plasma infused, right? And you put it in other containers and you can just drink from it. That's when I start knocking my shit, and I'll do not a knockoff, but give you reality. Something that really helps you. Just bring me glass jars, I can plasmatize it. $25 per plasmatation. That's all. The plasmatation is turn to this from this reactor, and as you see in the readings, it'll help you. As you see with the people. The proof's in the pudding. I'll show you something. Something other quiche people know, or see, or my wife hasn't paid attention to. Look, honey, I'll share something with you. If it picks up on the camera. Okay? Watch. We're gonna go in a circle. Which way does the stuff move? Right? Do you see the plasma?
Mm-hmm.
What is it doing? Some was going in counter rotation to which way movement I was doing, and the other was going in the opposite. Are you picturing this and seeing people? Because the camera's picking up. It's not a figment of my eyes. I'm just rotating the circle. But if I go back this way.
Yeah.
But if I go
Looks
this
like
way.
it's layers to it.
Here, hold it.
And one layer is
Stand up and hold it,
more
please. Stand up and hold it so I can show you an observation and show these people. Because you know, whenever I show you something, the quiche people fucking steal it.
hold out in front of you and just articulate in a circle. Your body is the pivot, and this spin in a circle. What is it doing?
Well, the plasma is still suspended.
In the center. But what is perpendicular to you counter-rotates to the opposite way that you go.
Yeah.
You understand? No, Keisha, let me talking about
Yeah.
this. None of this stuff. You understand?
Yeah.
So now that you've gone that speed, now stop. Stop. Now it flows the other way.
Yep. And despite of the plasma, gravity is now going up instead of going down, and its density, instead of going down and sinking to the bottom, still suspended.
Now rotate that way. Now stop.
The bottom is rotating counterclockwise, and this is just still.
Excuse me.
I'm looking at this big conglomerate.
Do you understand?
Yeah.
By you doing that, there's a fat standing wave hitting on this. And quiche people. Kelia.
The only way I got to describe it is like there's different organizing fields. Like if
Remember
you
I
have-
always describe in certain people that have the tardigrade dream after I talk with them? What do they see? They see that blockchain. Jamie's son seen that when he prophesied about this in the Paradise Fire.
Oh, Jeannie's son?
That's
Esteban?
why we're making this, and I'm showing this to you, because we're going to go help sister.
Let me see.
See that, folks? No trickery. No quiche method. It's real life. Our stuff.
Morning, everybody. Making a piece for Sister Jeannie Schumacher. I may send you to the shop before me, dear, so I can finish this. But I won't be but more half an hour behind you, so I can finish Jeannie's piece for her. I would be calling Gary, but I'm going to load everything in the car. I just want to finish Sister's pennant. What was it? Yes, sir. That's fine. Do the silver around the doors when you get there, since you're not going to be open until I get there. Do the coconut oil on the outside and on the inside handle, please. Copy the protocol that they copied for me. Now you understand that, don't you? Teacher's teacher, let's see. The kittens. The zika. I love you guys. Sister, a pennant. And 1 cup, 1 line. Real one. ZIKR, Zikr. Zikr water. Activation of the interstitial water. Your H3O2. Which is not motherfucking Imran Kish. He is not the seal of the prophets. Muhammad Rasulullah is. Saint Mahdi is the wisest of the prophets. And Saint Isa comes after Mahdi. Mahdi comes next. Isa is Isa. Jesus is coming. What the fuck is this guy talking about? He's the Antichrist. Wake up, people.
ألا بذكر الله تطمئن القلوب ألا يا الله الله
That's what I do while I wire up as I make all my stuff. Is zikr. Torch and holding with not no pliers like Quiche. Low torch and nano coating with what? My own plasma. My own phonons, anions, and ions. Now how's it gotta get to melt some glass? Barehanded, right?
بالله العلي العظيم. هو 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 هو
Call landlord in 10 minutes. Make a payment.
الله ونعم الوكيل، نعم المولى ونعم النصير، لا حول ولا قوة إلا بالله العلي العظيم. الله الله حق، الله الله حق، الله الله يا هو يا هو يا دايم، يا هو يا
There's clear glass.
هو يا دايم، الله يا هو يا دايم، يا دايم يا دايم يا دايم يا الله، يا دايم يا دايم يا دايم.
Chilean miners, saved by us. Allah's mercy.
يا حليم يا الله، يا حفيظ يا حفيظ يا حفيظ يا الله، يا حفيظ يا حفيظ يا حفيظ يا الله، يا لطيف يا لطيف يا لطيف حنان يا الله يا ديان يا ديان يا ديان.
Lots of water, honey. See how clear it is here in Cali? Showing you that they're sending stuff to clear the sky out, which means you're getting dehydrated. Brother Jeremy. I'm gonna be sending you one also. Live on that, brother. Reach out. Let me know what's going on. Why use VP882 and so forth? And so forth. This coronavirus is gastric.
The human gut can be the scene for devastating conditions such as inflammatory bowel disease, which arises through an improperly controlled immune response. The gut is often the body's first point of contact with microbes. Every mouthful of food is accompanied by a cargo of microorganisms that go on to encounter the mucosa, the innermost layer of the gut. Most microbes are destroyed by the harsh, acidic environment in the stomach, but a hardy few make it through to the intestine. The intestinal surface is covered with finger-like protrusions called villi, whose primary function is the absorption of nutrients.
Coronavirus.
However, these structures and the underlying tissues also host the body's largest population of immune cells.
Depleting your body of its nutrients.
Scattered along the intestinal mucosa are dome-like structures called Peyer's patches. These are enriched in lymphoid tissue, making them key sites for coordinating immune responses to pathogens whilst promoting tolerance to harmless microbes and food. The villi contain a network of blood vessels to transport nutrients from food to the rest of the body. Lymphatics from both the Peyer's patches and villi drain into the mesenteric lymph node. Within the villi is a network of loose connective tissue called the lamina propria, and at the base of the villi are the crypts, which host stem cells that replenish the epithelium. Finally, the epithelium, together with its thick overlying mucus, forms an important barrier against microbial invasion. Embedded within the matrix of the Peyer's patch is a mix of immune cells including T and B lymphocytes, macrophages, and dendritic cells. A key function of the Peyer's patch is the sampling of antigens, in this case mostly bacteria and bits of food. To facilitate this, the Peyer's patch has a much thinner mucus layer as well as specialized phagocytic cells called M cells, which can transport material across the epithelial barrier via a process called transcytosis. Finally, dendritic cells are able to extend dendrites between epithelial cells to sample antigens that are then broken down and used for presenting to lymphocytes. Sampling antigens in this way typically results in so-called tolerogenic activation, where the immune system initiates an anti-inflammatory response. With their cargo of antigen, these dendritic cells then traffic to the T cell zones of the Peyer's patch. Upon encounter with specific T cells, the dendritic cells convert them into an immunomodulatory cell called a regulatory T cell, or Treg. Defects in the function of these cells are associated with inflammatory bowel disease in both animals and humans. These Tregs then migrate to the lamina propria of the villi via the lymphatics. Here, the Tregs secrete a molecule called IL-10, which exerts a suppressive action on immune cells within the lamina propria and upon the epithelial layer itself. IL-10 is therefore critical in maintaining immune quiescence and preventing unnecessary inflammation. However, a breakdown in this process of immune homeostasis results in gut pathology, and when this occurs over a prolonged period and in an uncontrolled manner, it can lead to inflammatory bowel disease. Chemical, mechanical, or pathogen-triggered barrier disruption, coupled with particular genetic susceptibilities, may all combine to set off inflammation. Epithelium coming into contact with bacteria is activated, leading to bacterial influx. Alarm molecules released by the epithelium activate immune cells, and Tregs in the vicinity scale down their IL-10 secretion to enable an immune response to proceed. Dendritic cells are also activated by this environment and start to release key inflammatory molecules such as IL-6, IL-12, and IL-23. Effector T cells also appear on the scene, and these coordinate an escalation of the immune response by secreting their own inflammatory molecules, tumor necrosis factor alpha, interferon gamma, and IL-17. Soon after the effector T cells have arrived, a voracious phagocyte called a neutrophil is recruited. Neutrophils are critical for the clearance of bacteria. One weapon in the neutrophil armory is the ability to undergo a dramatic form of self-destruction called netosis. This leaves behind a jumble of DNA saturated with enzymes called a neutrophil extracellular trap. Although this can effectively destroy bacterial invaders and plug any breaches in the epithelial wall, it also causes collateral damage to tissues.
That's a cytokine storm.
Slowly the tide begins to turn, and the bacterial invasion is repulsed. Any remaining neutrophils die off via apoptosis, a non-inflammatory form of cell death, and are cleared by macrophages. Epithelial integrity is restored by replacement of any damaged cells with new ones from the intestinal crypts. Finally, Tregs are recruited once again to calm the immune response. Targeting the molecules involved in gut pathology is leading to effective therapies for inflammatory bowel disease.
ZP882, bacterial macrophage, macro.
Opportunistic and pathogenic bacteria that penetrate past the epithelial barrier, entering the bloodstream, are efficiently killed by macrophages. It is clear that the immune system is not ignorant of their presence of bacteria, because a vigorous IgA response directed against these microbes can be measured, and a significant proportion of this IgA is induced through a T-cell independent pathway. This high-speed video shows a macrophage chasing down planktonic bacteria. This is likely a segmented filamentous bacterium, as it appears to be attempting to escape capture. Eventually, the macrophage is able to phagocytize the bacterium. An optimally functioning immune system is required for such events to occur in the body. In the next video, we observe a different scenario. In the second video, you can see a red blood cell infected by mycoplasma with the characteristic bumps. Two macrophages are attracted to the infected cell. As one macrophage is aggressively moving towards the cell, it is joined by another macrophage. The second macrophage is immediately repulsed from the mycoplasma-infected red blood cell. The first macrophage makes another attempt from a different direction. Eventually, both macrophages move quickly away from the mycoplasma-infected red blood cell. Molecular mimicry of host molecules enables bacteria to display surface molecules resembling those of the host. This contributes to immune hyporesponsiveness to bacteria.
Why you don't
Recent analysis has revealed the presence of secretion systems in commensal bacteria. Previously, such secretion systems have only been described for pathogenic bacteria. Opportunistic bacteria also use analogous secretion systems to deliver effector molecules that subsequently mediate their evasion of immune surveillance mechanisms in the body. This typifies the interaction between microbes and immune cells at the cellular level in immune-compromised individuals, where immune cells are rendered incapable of protecting the body.
As easy as writing capital letter V, capital letter P, 882 in the glass, and cytokine storm. Interferon. That which gets released causes so much damage to the body. Causes so much damage. This stuff gets triggered. This is what's going on.
Why does round pizza come in a square
Virus
box?
that they've infected the world with, so you see it in other organs and so forth throughout the body and muscles. You understand? Chimeriums and interferon.
In the last video lecture, we covered cells of the immune system.
My mom has had to have
Cells of the immune system which are widely dispersed in our body. Some are circulating in blood, some are circulating in lymph, and some are resident in various tissues and organs. When these cells recognize a foreign invader, they interact with each other and work together to destroy or eliminate the pathogen. Now, the question here is how the communication among immune cells takes place. The communication within the immune system is mediated by small soluble molecules which are known as cytokines.
Get these people for genocide.
Cytokines are produced and secreted by a wide variety of cells. They act as intercellular mediators. Once released, these cytokines bind to specific receptors on the surface of other cells of the immune system. And these specific receptors are known as cytokine receptors. Once a released cytokine binds to the cytokine receptor, it generates an internal signal, and this signal alters the activity of the cell. As a result, a cell may prepare to divide, it can undergo growth and differentiation, or it can secrete its own cytokines. Let us now talk about some important properties of cytokines. Cytokines are hormone-like substances. That means they are signaling molecules.
means they're metal.
Cytokines are soluble molecules, and some of them are membrane-bound molecules. Chemically, they can be protein or glycoprotein.
Equation.
Their molecular mass is less than 30 kilodaltons.
Like the plasma.
Cytokines have short half-life. This also explains that mostly cytokines act over a short distance. That is, either signaling is autocrine or paracrine. By autocrine we mean cytokine acts on the same cell, and paracrine means cytokine acts on the nearby cells. Cytokines are effective at low concentrations. This is because of the specific high affinity cytokine receptors. This means cytokine receptors are sensitive to low concentrations of the cytokines. The important cytokines which are involved in signaling between cells of the immune system are interleukins, tumor necrosis factor, interferons, colony-stimulating factors, and chemokines. Interleukins, abbreviated as IL. In Latin, inter means between, and in Greek, leukos means fight. So, literally, they are the cytokines produced by one leukocyte acting on the another leukocyte. But cells other than leukocyte may also use interleukins. Till now, about 35 interleukins have been identified. They are named interleukin, IL, followed by a number which represents sequence in which they were discovered. For example, T cell secretes interleukin 2. Interleukin 2 binds to the cytokine receptor present on the same T cell. This is an example of autocrine signaling. Interleukin 2 results in growth and activation of T cells. T cells also secrete interleukin 4. Interleukin 4 binds to the cytokine receptor present on the B cell. And then interleukin 4 results in differentiation of B cells into plasma cells. Tumor necrosis factors, abbreviated as TNF. Tumor necrosis factors are produced by mast cells, macrophages, and T cells. They are often firmly anchored into the cell membrane. So, tumor necrosis factors are those cytokines which are membrane-bound. They regulate immune responses and inflammation. They are responsible for apoptosis of many cell types. For example, macrophages release tumor necrosis factor alpha. Tumor necrosis factor alpha binds to the cytokine receptor present on the neutrophil and result in activation of neutrophils. Interferons, abbreviated as IFN. Interferons are so named because of their ability to interfere with the viral replication. They are of two types, type 1 interferons which comprises of interferon alpha and interferon beta. These are produced in response to viral infections. Type 2 interferons comprises of interferon gamma which increases phagocytosis by macrophages. Let us see how interferons provide antiviral defense. When a virus infects a cell, it induces the infected cell to produce interferons. These interferons are released by this cell and they bind to nearby uninfected cells. Uninfected cells now produce antiviral proteins. When the virus comes to infect this uninfected cell, the antiviral proteins of the cell degrade viral nucleic acid and inhibit protein synthesis. Thus, interferon prevents spread of viral infections. Colony-stimulating factors, abbreviated as CSF. Colony-stimulating factors are essential for the growth and differentiation of immature leukocytes in the bone marrow. that is, red blood cells, monocytes, granulocytes, and lymphocytes. Colony-stimulating factors ensure that the body is supplied with sufficient white blood cells of all types. Thus, this supply will determine the immunity of our body. Let us look at the important colony-stimulating factors. MCSF, monocyte colony-stimulating factor. MCSF is produced by T cells and bone marrow, and they affect the growth of cells of the monocyte lineage. GM-CSF, granulocyte macrophage colony-stimulating factor. GM-CSF are produced by T cells and macrophages, and they help in growth and differentiation of dendritic cells. GCSF, granulocyte colony-stimulating factors. They are produced by monocytes, and they help in differentiation and development of neutrophils. Chemokines. Chemokines acts as chemoattractants, and they signal leukocytes to move. Thus, chemokines control the movement of leukocytes. For example, when there is an infection, the chemokines secreted at the site of infection signal the leukocytes present in the blood, such as neutrophil, to migrate at the site of infection. In such cases, neutrophil become the principal phagocytic cells at the site of infection. So, now we understand that communication within the immune system is mediated by cytokines.
So, what the hell is the Quiche Foundation doing? Not what I'm doing. They're supposed to be the space, and they got all these people infected around the world, and smart and all that, right? I don't get it. Sister that I'm making the pennant for. Work for the city. Picked up the cats. The people she picked the cats up from, they're friends with people that just came back on a cruise ship. Following Keisha's plan. Come back on a cruise ship and infect people, right? People. Not to my friends, not to my community, not to our community, not to our friends.
ونعم النصير. لا حول ولا قوة إلا بالله العلي العظيم. بسم الله الرحمن الرحيم. بسم الله